Hello friends, welcome to online biology lectures 2020. I hope that you are all doing good. My today's video is about the nomenclature of enzymes. You all know that enzymes are the biological catalysts that play a role in speeding up the chemical reaction. In the absence of the enzymes, the reaction may take several hours or it may proceed very slowly. So enzymes play their role by increasing the rate of reaction 10 raised to power 3 or 10 raised to power 8 folds. We have discussed about the classification of the enzymes. Enzymes are classified into six major classes that include oxidoreductases, transferases, hydrolases, liases, isomerases, and ligases. Now we will discuss about the nomenclature of the enzyme. Each enzyme is assigned two types of names. One, the recommended name that is convenient for everyday use, and the second name that is more complete and with which the enzyme can be identified without any ambiguity. That is called the systemic name. So first of all, we will discuss about the recommended name. We all know that the name of an enzyme ends with a suffix called ase. These are the most commonly used names for the enzyme. It depends upon the substrate to which it is attached, to which it binds and converts into a product. For example, glucosidase, urease, sucrase, etc. As the name suggests, urease, it shows that it acts on the urea. Sucrase acts on sucrose, glucosidase acts on the glucose. Sometimes there is further description added in the recommended name. For example, lactate dehydrogenase, that is, it acts on the lactate and helps in the removal of a hydrogen, and adenylate cyclase, etc. But it also be noted here that some enzymes retain their original white trivial names, for example trypsin, pepsin, etc. The other name is the systemic name. The International Union of Biochemistry and Molecular Biology IUBMB developed a system of nomenclature in which the enzymes are divided into six major classes, as we have previously discussed. The suffix "ase" is attached to the end of each class, which completes the description of a chemical reaction catalyzed by these enzymes. For example, D-glyceraldehyde 3-phosphate, NAD reductase, etc. The IUBMB names are unambiguous and more informative, but sometimes they become more complex to be of the general use. Here you can see the skeletal outline of IUB system. Each of the six major classes of the enzyme has 4 to 13 subclasses depending upon the type of the reactions they catalyze. Each enzyme name has two parts. The first part constitutes the name of the substrate, and the second name ends with the suffix ase and it shows the type of the reaction that is catalyzed. Any type of additional information, if needed to clarify the reaction, may follow parentheses. This reaction is showing a type of a decarboxylation reaction which is followed by the removal of a hydrogen. It belongs to oxidoreductase class, that is the first class of enzyme classification. Here 1.1.137, it designates the class, major class, subclass, subclass of the enzyme. The enzyme commission code with the first character shows the type of reaction, the type of class, the second digit shows the type of subclass, the third digit shows subclass, and the fourth digit is for the specific enzyme. So enzyme commission 2711 denotes class 2, that is transferases, the major class, subclass 7, that shows the transfer of phosphate, subclass 1, that it is an alcohol, that is a phosphate acceptor, and the final digit denotes the hexokinase or ATP dehexose 6-phosphoryltransferase. which means it is an enzyme catalyzing phosphate transfer from ATP to hydroxyl group on the carbon 6 of the glucose. So the enzyme commission number helps in determining the major class, subclass, subclass and the type of reaction, the type of substrate on which the enzyme is acting, and it helps us to determine the overall information of the enzyme. Now comes the structure of an enzyme. We know that every enzyme has a special site to which a substrate binds and is converted into the product. That site is called the active site of the enzyme. It contains special pocket or cleft. The active site, it contains amino acid side chains that create a 3D surface complementary to the substrate. The substrate binds to the active site of the enzyme, forming a complex called enzyme substrate complex. Enzyme substrate complex is then converted into enzyme and the product. The enzyme is released and the substrate is converted into the product, which subsequently dissociates. The enzyme product complex subsequently dissociates to enzyme and the product. Some enzymes need a non-protein part for their functioning and for their activity. That non-protein part is called cofactor. Commonly encountered cofactors include metal ions, for example zinc and ferrous, or any organic molecules called coenzymes that are often the derivatives of the vitamins. If an enzyme do not need any cofactor, if an enzyme do not contain any cofactor, that enzyme is called holoenzyme. The protein part of the holoenzyme is called apoenzyme. In the absence of appropriate cofactor, the apoenzyme typically does not show biologic activity. A tightly bound coenzyme that does not dissociate from the enzyme is called a prosthetic group. Most of the enzymes can contain additional active sites for interaction with regulated molecules called the allostatic sites. Enzymes that contain the allostatic sites are regulated by the effector molecules called the modifiers or the modulators that are non-covalently bound to the site that is other than active site and called allostatic site. Presence of the allostatic site affects the affinity of enzymes for its substrate, or it modifies the maximal catalytic activity of the enzyme. Effectors that negatively regulate the activity of enzyme are called negative effectors, whereas those effectors that increase the activity of enzyme are called positive effectors. As we have previously discussed that enzymes are specific for the type of substrate they catalyze. The specificity of enzyme is determined by the functional group of the substrate, the functional groups of the enzyme, and the physical proximity of the functional groups. You all know that there are two types of theories that explains the interaction between the substrate and the enzyme active site, that is the lock and key model and the induced fit model. We will briefly discuss both of these two theories here. Number one, the lock and key theory. It states that the enzyme active site is complementary in conformation to the substrate. So the enzyme and substrate recognize one another just as a key fits into the lock. The shape of the substrate is just like the shape of the active site of the enzyme. As you can see in this picture, the substrate is shown in the red color and the enzyme is shown in the blue color. You can see that there is complementarity between the active site of the substrate and the active site of the enzyme and the substrate, so it easily fits into the active site so the substrate easily fits into the active site of the substrate of the enzyme. The other theory, called the induced fit theory or induced fit model, it states that the enzyme changes its shape depending upon the substrate, means the shape of the substrate and the shape of the active site of the enzymes are not complementary, so the enzyme has to change the shape of its active site so that substrate can fits into it. So the conformation of substrate and enzyme is only complementary after the binding. So when the substrate binds to the active site, the active site changes its shape and it becomes complementary. You can see that in the presence of a substrate, the active site undergoes a change in its conformation to allow a better fit between the active site and the substrate. This means that enzymes are highly specific for the reaction they catalyze. You can see in this picture the substrate has a different shape and the active site of enzyme has a different shape. So when the enzyme substrate complex is formed, the enzyme has undergone a little change in its active site so that the substrate can easily fit into it.